Hello, and welcome to Antibiotic Potential of Flathead Fungi and Flora by Terri Ridenour, Kate Smith, and Ethan Harbo from Flathead Valley Community College in Kalispell, Montana. My name is Terri, and I will give you a little bit of background on our research. As of 2018, only 4 brand new antibiotics had been developed in the last 40 years. This is concerning because 2.8 million people in the US suffer from an antibiotic-resistant infection every year. Plants and other organisms may have the potential to help this ongoing crisis. Our primary research objectives are to collect and identify plant, fungi, and lichen species from the Flathead Valley, and to extract secondary compounds from those species using different solvents, and ultimately testing those extracts against different strains of bacteria and yeast. Originally, the focus of this research was to only collect lichen and determine their antibiotic potential. That got expanded to include any plant or fungi in the Flathead Valley. For the last 2 semesters, the focus has been on plants and fungi with any recorded traditional antibiotic uses. However, we will collect anything that looks interesting because we have had some promising results from plants and fungi that have not been previously researched for antibiotic potential. After the samples are collected, they are brought back to the lab, identified if needed, and given an identification number. The samples are then prepared for storage by dehydrating the ones that need it and are placed in envelopes with their identification number. The samples are then stored in our herbarium until they are used in one of our 2 extraction methods. Both extraction methods use the same 3 solvents with different polarities in the hopes of pulling out different secondary compounds. Here's Kate to tell you more about secondary compounds and the extraction process.
Hi, my name is Kate Smith, and these are secondary compounds. Secondary compounds exist in virtually all plants, fungi, and lichen. They are not necessary for primary functions like plant growth or photosynthesis, and they are primarily used as defense mechanisms. For example, the latex in a poppy plant is used as a defense mechanism against herbivores and pathogens. Morphine, found in the latex, is used as a pain medicine by humans. The compound nicotine is produced as an insecticide by the tobacco plant and is the active ingredient in cigarettes. Bread mold results from spores landing on the bread and forming a colony. The antibiotic penicillin is derived from this occurrence. Our first method of extraction involves using a mortar and pestle to pulverize sample tissue and combining that with 25 milliliters of the solvent. It is then agitated on a shake table for 24 hours. After that time, the extract is pipetted into small tubes, centrifuged, and is then pipetted again to separate out the plant material into fresh tubes. Those tubes then go into a vacuum desiccator and run until all the liquid has been desiccated off, and what is left is a small, dry pellet of plant material. The samples are then placed into a negative 80 degrees Celsius freezer to wait for testing. Our second method of extraction involves using a sock slit extractor. This method allows us to use whole or pulverized tissue samples. 2 grams of the plant tissue and 190 milliliters of the solvent cycle through the sock slit for 15 to 20 hours. After the extract is removed, it is divided equally into 6 large tubes and placed into a large vacuum desiccator until the amount of extract reaches 5 milliliters. The extract is then pipetted into small tubes. Those tubes then go to a smaller vacuum desiccator and run until all the liquid has been desiccated off and what is left is a small dry pellet of plant material. These samples are also placed into the freezer to wait for testing. This last picture demonstrates how different solvents can extract different compounds from the same plant tissue. Now here's Ethan to tell you about testing.
My name is Ethan Harbo. If you'd like more specific information about the testing process, check out Antibiotic Potential of Lichens, Fungi, and Bryophytes in Northwest Montana. They will be presenting on Monday at 3 p.m. Eastern Time, session number 2589. We'll give a brief overview of the testing right now. Desiccated samples are resuspended and tested using the Kirby-Bauer disc diffusion method against 4 gram positive, 3 gram negative, and 1 yeast. A zone of inhibition greater than 6 millimeters is considered a positive result. The samples with a zone of inhibition greater than 6 millimeters undergo further testing and are tested for minimum inhibitory concentration. Since the beginning of 2019 until now, 38 different fungi, lichen, and plant species have been tested. Of those, 21 species inhibited bacterial growth. Amanita muscaria, which has little research done on its antibiotic properties, inhibited the growth of all 4 gram-positive bacteria. Lyophilum decastes, the fried chicken mushroom, was one of our few extracts which showed antimicrobial activity against Candida albicans yeast. Our upcoming goals include desiccating extracts that have been prepared and have been stacking up in the freezer, compare and contrast the differences between whole and pulverized tissue extracts, compare and contrast the role different solvents have in extract efficacy. And one of the final steps of our research is to determine the identities of the active secondary compounds that are inhibiting bacterial growth. A local analysis lab has offered to assist in LCMS and GCMS analysis and identification. This would be a huge step because it would allow us to see if any novel compounds are present in the extracts we have made. Another goal is to finish our lichen and fungi field guides. These will be used as identification guides for future research students and the public. We'd like to thank our mentors, Dr. Mirbai McCarthy and Dr. Ruth Reitzman, current and past researchers, Montana INBRE and the FVCC Foundation, Tim Wheeler at University of Montana, Annie Beal, our biology lab coordinator. Thank you for listening to our talk. If you have any more questions about our research, you can come view our presentation, number 5630, 5 p.m. to 6 p.m. Eastern Time. Thank you.